Hi, everybody. So welcome back to the last part of our intro to nervous system. So last time we talked about the graded potential and how graded potential may or may not lead to action potential depending on the ligand channels and the frequency of the ligand-gated channels that are open. So we are going to kind of dive in now to see how graded potential may potentially lead to action potential in a little bit more detail, and then we'll talk about the action potential itself. So in this scenario, and I'll show you this picture at the end of the previous PowerPoint to just kind of give you an idea, and we're gonna go into a little bit more detail about this now. So imagine this is your presynaptic axon ending at your axon terminal, and this is your postsynaptic neuron on the bottom. So what you will see is if this neuron is activated and there's an action potential coming down here, it will eventually reach the axon terminal. Now in the axon terminal, you have these channels right here, which is labeled as calcium. These are actually calcium voltage-gated channels that are basically going to correspond to your action potential of your axon. So under normal condition, these calcium channels are closed. Now, when they are going to notice the electrical impulse or the action potential of the axon, these calcium channels are going to pop open and allow the calcium to rush in. Again, remember that calcium concentration is high around the cell, which means when the channel is open up, calcium goes from high concentration to low concentration. Now, what is the importance of these calcium channels here? When calcium comes in, it allows these vesicles that carry neurotransmitters to be able to move, go to the end of your axon terminal, and allow the neurotransmitter to be released into the synaptic cleft of your neuron. So without this calcium, there is no exocytosis of your neurotransmitter into the synaptic cleft. So if I ask you again, what is the purpose of calcium? Is to basically allow the exocytosis of neurotransmitter into the synaptic cleft. Now, as I said before, these these neurotransmitters can go and bind to the ligand-gated channels, the three types that we described in detail. And when that channels open up, depending on the channel, you're either moving in sodium and potassium, moving sodium in and potassium out. You're either moving chloride in or you're moving potassium out. So depending on with these channels, you can create an depolarization effect or a hyperpolarization effect. So let's go ahead and move on to my beautiful drawings. So electrical impulses, again, as what I just described, causes the presynaptic neuron to open up its calcium voltage-gated channels. Influx of that calcium causes the neurotransmitter to be exocytosed from its vesicles into the synaptic cleft. And at that point, that neurotransmitter can either cause two scenarios: what we have described as a depolarization effect or what we have described as a hyperpolarization effect. Now, the proper terminology for that, based on the synaptic region, is if you have a depolarization effect, it's called excitatory postsynaptic potential, or EPSP. Sorry, I wrote that as ESPS. EPSP. And that depolarization is a result of sodium-potassium channels. Or you can have inhibitory postsynaptic potential, which is IPSP, created by either chloride channel moving chloride in or potassium channels moving potassium out. Here is the depiction of what we're describing, guys. So again, you're starting with that -70. So hopefully everybody remember, -70 is your resting membrane potential. If you have the sodium-potassium ligand-gated channels open up, you will have depolarization, as in your cell is becoming positive. And if it reaches the threshold, you will see an action potential forming up. That is excitatory or EPSP. In other scenarios, if you have chloride channels opening, chloride coming in, making your cell more negative, or potassium leaving your cell, making your cell negative, then you have a hyperpolarized scenario or hyperpolarization. And this is known as IPSP or inhibitory postsynaptic potential. So what are these cases or scenarios that I've described where you have many neurons or many stimulation coming in together? What are the possible scenarios? So the simplest form or scenarios that we have is when you have kind of a 1-to-1 ratio, or not necessarily 1-to-1, but all of the signals being excitatory, right? So the signal in this case is excitatory. Okay, so E1 implies excitatory postsynaptic potential. Notice that the signal is coming in, but you are failing to reach your threshold because the original stimulation, this red arrow right here, the original stimulation is not strong enough or frequent enough to cause an action potential by reaching a threshold. So you have one, it degrades, you have another stimulation, you degrade. So in this scenario, you have no action potential, no activation of your neuron. Now, in the same scenario, what if you have the same neuron creating an excitatory postsynaptic potential, but the stimulation is more rapid? So you basically continuously causing the postsynaptic neuron to be excited, okay, to become depolarized. So in that case, we have what we identify as temporal summation. When you have multiple excitatory forces come in, so even if the first one didn't reach your threshold to create an action potential, it doesn't go back down completely to -70. As the second excitation comes in, that helps you depolarize enough to reach your threshold and create your action potential. Now, what if you have what we identify as a spatial summation? Spatial summation can be when you have either two identical forces affecting a neuron or opposite forces, excitatory and inhibitory, affecting your neuron. So in this scenario, you have excitatory scenario 1, a stimulation from one neuron, and a stimulation or excitation from another neuron. So both of these can basically create a very strong stimulation that causes the depolarization to reach the threshold, and you will see an action potential. However, what if I have a spatial summation, but it's two forces working against each other? If you have excitatory and inhibitory working against each other, what I've described before. For instance, you have sodium potassium channels depolarizing, and at the same time you have your chloride channel hyperpolarizing. So if I had normally just the inhibitory force, let's say chloride channels, I will hyperpolarize and there is no chance of me creating an action potential. What if I have two forces coming in together? Now in this case, the excitatory was bigger than the inhibitory, so I went slightly above, or I have a slight depolarization, but that depolarization didn't reach the threshold. So let me just kind of introduce another concept to you. So what if I have another excitatory here and an inhibitory? So in this scenario, the chances are that these two excitatory force is big enough for me to what? Reach the threshold and create my action potential. So it depends on which of these forces, call them good and evil, are actually stronger. If the excitatory is stronger, there is a chance of action potential. If the inhibitory force is stronger, you're always going to hyperpolarize and take away that chance of action potential. How do I end degraded potential? How do I basically cause my ligand-gated channels to stop moving and opening the ligand-gated channels? You can do that by one of the following. You can either reuptake your neurotransmitter back into your presynaptic neuron, so reclaiming what you have basically in the synaptic cleft. You can also break down the enzyme within the synaptic cleft. So when the enzyme is not the original structure, then it cannot open your ligand-gated channels. Third scenario is when your neurotransmitters literally diffuse away from your synaptic cleft. Again, this is the idea of diffusion, right? So in a synaptic cleft, you will have really high concentration of these neurotransmitters, which the physical law allow them to start to diffuse into areas with a lower concentration. If the concentration of the neurotransmitter is low enough, then they cannot really open too many of the ligand-gated channels, and therefore they cannot create an effect on your postsynaptic neuron. Now we have talked about graded potential, reaching the threshold, which is -55, and the fact that it can create an action potential. In a previous lecture, I also mentioned that the action potential relies on your voltage-gated channels. So these voltage-gated channels are going to be your sodium VGCs and separately your potassium VGCs. So kind of introducing the concept for you here. Depolarization is going to highly depend on your sodium VGC, basically moving a significant amount of sodium in. Repolarization is going to depend on your potassium VGCs, and same thing as your hyperpolarization. Resting membrane potential is basically, we have talked about it many times already, but that depends on your sodium, potassium, ATPase, as well as your sodium leakage channels and potassium leakage channels. If this information seems a little bit unfamiliar, please go back and review your previous PowerPoints for this information. So right here, we have figured out that you have reached the threshold, so we're not really discussing that anymore. We're just gonna focus on these steps right here. So depolarization. During depolarization phase, your sodium VGCs are gonna open if you have reached the threshold of magic -55 millivolts. Sodium is gonna influx and cause the interior of the cell to become less negative and actually move it all the way to +30. So look at your picture. You're going basically from -70 up to the threshold, which is maybe not depicted as well here, and then eventually reaching a +30. So at this peak, right here, there's a couple of changes that are going to happen. One is your potassium VGCs start to open. And then the main thing that is happening is your sodium VGCs are going to go inactive, guys. Not closed, but inactive. And I'll talk about that, why that's so important. So during repolarization, sodium VGCs go inactive first, right? And then potassium VGCs open. So when potassium VGCs open, you have a lot of potassium outside, and these potassiums are start to move out of the cell. So you're starting to become negative. So you just kind of go down, right? Until you reach back to -70. Now again, during this repolarization, another thing is also happening. So as the repolarization happened, not at the top, at this phase where the repolarization happened, your sodium VGCs are also going to go from inactive to closed. So pay attention to this. I'll come back to it in just a second. Hyperpolarization, which is when you dip farther than -70, you become even more than -70. The reason for that is because potassium channels that you have that causing the repolarization are opening at different times, and therefore they're going to close at different times. So the one that are opening somewhere up here, okay, they're gonna close somewhere down here. But if you open up the potassium channel somewhere here, they're gonna close somewhere down here. And again, the more potassium you lose, the more negative you become. And because you're continuously losing potassium, you can get pretty negative or very close to the equilibrium for the potassium level, which is around -90. So you reach about -85. And by the time you get to -85, all of your potassium channels would be closed, and therefore you have the chance to go back and reset back to the membrane potential using your sodium potassium ATPase as the main channel. So kind of think about it. During the depolarization, you brought a lot of sodium in, right? Now sodium potassium ATPase is going to take that back out. During repolarization and hyperpolarization, you took a lot of potassium and took them out of the cell. So sodium and potassium ATPase is not going to put that potassium back into your cell. So literally kind of like, I think about it as the rides in your like Disneyland or Universal, where you have everything like breaks apart, right? And then they put everything back to its place, and then they redo the process. So when you break it down, that's your like depolarization, repolarization, hyperpolarization. You're putting everything back to where they belong. That's your sodium potassium ATPase. So let's quickly talk about refractory period. So there is something called refractory period, where basically you either have no capacity to create a new action potential, or you can create an action potential, but you need a lot of stimulation. So absolute refractory period, which is kind of highlighted in this picture in green, is where your sodium voltage-gated channels are either active or open, which means you cannot open them anymore, or they are in an inactive phase, which means you cannot open them. They're already in an open position, they're just inactive. They're using the inactivation gate. Why is that important? So this is important because you want the electrical impulse to always move from the axon hillock to the axon terminal in one direction. But imagine if these channels that are located in the region get stimulated or receive another stimulation from another graded potential. You potentially can have an action potential that's moving down the axon terminal, and another action potential that's moving toward your cell body. And this is a big no-no. You basically only want to have a unidirectional electrical impulses. So by keeping your sodium voltage-gated channel inactive, you basically limiting the chance of creating a new action potential, possibly going in the wrong direction. Now, as the inactive channels start to become closed, so some of these channels are starting to close, then at that point, you're going to say, oh, okay, there is some closed channels that can be reopened, right? Which means that you're going to create the same action potential in the same direction. However, for this situation to happen, which is called a relative refractory period, you really need to put a very strong stimulation that your axon says, okay, I need to do this really. I need to create a new action potential. So that's relative refractory period. When your cells or your sodium channels, sodium voltage gated channels are slowly switching from inactive to an closed phase. Please do keep in mind, absolute refractory period, you have no chance of creating a new action potential because those sodium voltage gated channels are either already open or they are in an inactive phase. So you are really cannot create a new action potential regardless of the amount of stimulation you're receiving. Here's the same depiction. Pictures are sometimes a little bit off, but absolute usually covers part of the repolarization, and then the relative refractory period, as I've showed you earlier, covers part of the repolarization and the hyperpolarization. When you go back to the resting membrane potential, basically everything is where it's supposed to be. Your sodium VGCs are all in a closed phase, which means you can easily restart the whole process and create a brand new action potential. One of the last topics I want to talk to you guys is the type of action potential that we create. So the action potentials we create does not change. It doesn't matter how strong your stimulus is, the action potentials do not change. So you always go from -70, you reach the threshold at -55, and the maximum you're gonna reach is +30. So the question is, well, what if I have a super strong stimulation versus a really weak stimulation? How do I distinguish these two? What you're distinguishing them, or how you're distinguishing them, is how many action potential you will be creating as a result of that stimulation. So let's look at this scenario. You have a scenario where the graded potential does, or the stimulation that leads to graded potential is not even strong enough to reach the threshold, right? So you have no action potential up here. What if you have a stimulation that goes a little bit above the threshold? Then you get relatively frequent stimulation or action potential, but again, they're kind of spaced out here, right? So you have one, then another, then another, then another. Now, the third scenario is you have really a strong graded potential as a result of a very strong stimulation, where it's pushing you significantly higher than the higher than the threshold. So in this scenario, you're still gonna create the same action potential. You're just gonna do it a lot more faster. So all of your action potentials become super close together with very limited space between, in terms of time between them. So kind of highlighting this, it may be very unclear if you're just kind of thinking about it in the other sense. So this line that goes up, regardless of which one I'm talking about, that both represents depolarization and repolarization. And the little ones that go below 70, that's depicting your hyperpolarization. So a lot of my students say, well, I only see one line. So basically what I'm saying is, if we kind of want to clarify what I'm talking about, this line, if I kind of break it and open it up, this is what it would look like actually, right? So this peak you see is from here to here, and the hyperpolarization portion would be down here. So that was about it, guys. So keep in mind, again, the difference between IPSP, inhibitory versus excitatory. What scenarios of graded potential can possibly lead to an action potential? And then what are the steps of action potential? What is the difference and the purpose of absolute versus refractory period? And then lastly, how different stimulation actually affect the action potential. So that's about it for this PowerPoint. Thank you, guys.